All right, here we go with DNA structure, which should be completely and totally review. All right, so I'm just going to kind of jet through it. So we know that DNA is made of genes, kind of continuing from the previous screencast. But how exactly, it's a very simple molecule when you look at it. So how exactly does it convey inheritance? How does genes do what they do? So we're going to get into that a little bit. We're going to start with the structure of DNA, because if we want to know what the function of DNA, we have to know about the structure, because again, structure and function. I say structure, you say function. Structure. Well done. All right, DNA for different nucleotides. Each nucleotide has 3 components: a phosphate group, a deoxyribose sugar, and 1 of 4 nitrogenous bases: thymine, cytosine, adenine, and guanine. And if you remember again, thymine and cytosines are pyrimidines, and adenine and guanine are purines. Okay. Here they are again. So these are my purines over here. And over here are my pyrimidines. And you'll notice the pyrimidines have the nitrogenous base is 1 ring. Okay. And for the purines, the nitrogenous base is made of 2 interconnected rings. Other than that, the 4 are the same. The sugar and phosphate part is exactly identical. And if you remember, what is going to be key while we are here is this 5' phosphate. It's on that 5' carbon. And this 3' hydroxyl. Okay, that is what is going to be used to form a bond with the next nucleotide. The 5' phosphate bonds to the 3' hydroxyl, and we get a string of nucleotides and make our nucleic acid. And that's basically actually DNA replication in a nutshell. If you remember, our good friend Chargaff came up with the rules of base pairing. He noticed that there were equal amounts of adenine and thymine and equal amounts of guanine and cytosine. Okay, so G pairs with C and A pairs with T. DNA is actually a double helix of 2 strands of nucleotides. This was proved by a couple of other scientists. Some you probably have heard of, some you may not. Here's Rosalind. Oh, R.I.P. Rosalind Franklin. My poor little hamster. Oh, I cry. But anyway, Rosalind Franklin was the bomb. And she and her boss, Maurice Wilkins, studied the structure of DNA using X-ray diffraction. And she created this incredibly famous photo, which we're going to see in a minute. And basically, you can extract information about the structure from the X-ray diffraction pattern picture that you get. So Wilkins and Franklin deduced the following information about DNA. Okay, it's long and thin, uniform diameter of about 2 nanometers. It is a helix, which means it's twisted corkscrew or like a spiral staircase. Okay, so that was point number 2. Point number 3, it's actually a double helix. There are 2 spiral staircases kind of going in. Okay, that was bad. Let's try again. There we go. There we go. 2 staircases kind of spiraling in opposite directions. It's made up of repeating subunits, just the 4 nucleotides over and over and over. And the phosphate groups were probably on the outside of the helix. And this, we're getting a little bit better idea as to kind of that ladder form. And this is the very famous photo. It's called Photo 51. And I will probably show you guys, there's a great movie called The Secret of Photo 51 that talks about the race to discover the structure of DNA, which these guys were all a part of. All right. And this X in the center is the signature of a helix. Our friends Watson and Crick, these are probably the ones that you've heard of. They were buddies with Wilkins, who was Rosalind Franklin's boss. And they were sort of ish working together. Wilkins was working with them and they were stealing Rosalind Franklin's data. So working together is kind of a loose term here. Okay. So again, these guys proposed that there was a single strand of DNA. It's a long polymer of many repeating subunits. With each DNA strand, the phosphate group of one nucleotide bonds to the sugar of the next. Again, that 5' to 3' bonding. Okay. And this gives us our sugar phosphate backbone. Okay. Then the nucleotides protrude from the backbone. Here's my backbone, my sugar phosphate backbone. The nucleotides protrude towards the inside. Okay, so if I have, so this would be one strand. If I had another strand over here, they would protrude in as well. And then these guys hook up, complementary base pairing. Okay. All right, and and again, let me go back to this. So this little dots that I drew between the base pairs are, of course, our good friend hydrogen bond, because hydrogen bond is the answer to everything. All right, so hydrogen bonds between complementary bases hold the DNA strands together in this double helix. Okay, so our DNA model is 2 DNA strands assembled like a twisted ladder. Bases protrude inward, and the sugar phosphate is like the side rungs, and hydrogen bonds hold the base pairs together. Okay. Cool thing about DNA is the DNA double helix, the strands are said to be anti-parallel. They're oriented in opposite directions. So if I have my ladder here, my base is in the middle. If this strand goes 5' to 3' over here, okay, then the corresponding strand is parallel, but in the opposite direction, 5' to 3. And we always read 5' to 3'. Okay. Because of their structures and the way they face each other, right? A bonds with T, thank you, Chargaff. G bonds with C, thank you again, Chargaff. Okay, this is called complementary base pairs. So if you have any sequence of DNA, you can get the corresponding sequence. Easy as pie. Okay. Complementary base pairs exchange Chargaff's rule. Okay. This basically, again, is saying A goes with T and G goes with C. Adenine and guanine are my purines, and thymine and cytosine are my pyrimidines. They're smaller, again, because there's just one ring. The purines are larger. They have 2 rings. And base pairing always puts a purine with a pyrimidine. Okay, we look back here. Again, adenine and guanine are purines, and cytosine and thymine are pyrimidines. So we always base pair a purine with a pyrimidine. And so there's always a large with a small. And that gives the constant diameter of the helix. Okay. And in 1953, Watson and Crick consolidated all of the data they had about DNA and the data they stole from Rosalind Franklin, don't even get me started, into the first accurate model of DNA. And there it is. Okay, so again here, if we have our phosphate at this end, that would be our 5' end. There would be a 3' hydroxyl at this end. So we go 5' to 3' in that direction. The opposite strand is anti-parallel, 5' phosphate to my 3' hydroxyl sugar there. Okay, and again, we see the nitrogenous bases sticking into the center. So our sugar phosphate backbone, the base is sticking into the center and base pairing up. We always see a purine with a pyrimidine, pyrimidine with a purine, purine, pyrimidine, pyrimidine, purine, etc., and so on. Okay. This picture here in the center shows that lovely twisted helix a little bit better. And then here's a nice ball and space filling model. Okay. All right. And I think that is all. Thus endeth the review. Got it? I hope so. Moving on.